Hello everyone, in this video we are going to discuss about N-linked glycosylation. So we have to understand where is this N-linked glycosylation taking place. It is taking place in the endoplasmic reticulum, and inside the lumen of the endoplasmic reticulum it is taking place. So why it is taking place? As the proteins are synthesized, they need to be transported inside the lumen of the endoplasmic reticulum, lumen of ER, so that the proteins need to be modified after the synthesis. So the modification takes place inside the lumen. So as we are telling, it is N-linked glycosylation. We may think that suppose it is a protein and the proteins are having N-terminal region and the C-terminal region. The modification may take place in the N-terminal region. Modification may take place in the N-terminal region, but it is not true. Where the modification is taking place, it is taking place in the proteins which are synthesized and transported inside the lumen of the endoplasmic reticulum, and in specific sequence only the glycosylation will take place. And the sequence is known as aspargine, and besides the aspargine there will be X and serine or threonine, and this X can be any of the amino acid except proline. Okay. So, as we are discussing here about the glycosylation, what is glycosylation? Glycosylation means addition of or transfer of oligosaccharides. And these oligosaccharides are of 14 types. So, what are these 14 types? Of the 14 types, there are 3 glucose, 2 N-acetyl glucosamine and 9 mannose. These are the 14 oligosaccharides which need to be transported in the protein and in some specific sequence, that is in the aspargine. That is the reason it is known as N-linked glycosylation. So, if these oligosaccharides are transported into the protein, it needs a enzyme, and the name of the enzyme is transferase. Oligotransferase. Oligotransferase is the enzyme which will help in the transfer of this oligosaccharides into the protein, and this oligotransferase is a membrane bound protein, membrane bound enzyme. It is a membrane bound enzyme and it is present near the lumen site. So, as it is present, as the enzyme is present near the lumen site, the modification will take place inside the lumen. So, for this addition, first of all the oligosaccharides need to be activated, and for the activation we need some of the nucleotides. We need nucleosides. What may be that? They may be UDP, GTP, CTP and many more. Let us see with the help of this diagram what it is taking place. So, for this process to be done, we need another lipid. What is the name of the lipid? The name of the lipid is dolicol, and this dolicol is a membrane bound lipid. It is a membrane bound lipid. Okay. This dolicol, first of all 2 phosphates should be added upon this. This first phosphate is added with the help of CTP when it is converted into CDP. Here 1 phosphate is added. Okay. So, 2 phosphate needed to be added upon this dolicol. After that only the oligosaccharides can be added. So, what happens? Here 1 phosphate is added through the help of the CTP when it is converted into CDP, because we get 1 phosphate from it, because 1 phosphate is released when CTP is converted into CDP, and this process takes place inside the cytosol. What takes place inside the cytosol? The activation of the oligosaccharides takes place in the cytosol. Okay. For that we need UDP, GDP. Okay. So, what happens? See, in this case, in the UDP, 1 N-acetyl glucosamine is added. That means UDP catches one N-acetyl glucosamine and it transfers into the dolicol. So here we are having UDP, here we are using UMP. UDP is converted into UMP. That means one phosphate is released. This phosphate is here. Here we are having one phosphate and an acetyl glucosamine is placed over here. In the next step, we know that we are using 2 N-acetyl glucosamine. So in the next step, another 1 N-acetyl glucosamine will be added. Say here we are having only one glucosamine, N-acetyl glucosamine. Here by the help of one UDP, attached to it there is one more N-acetyl glucosamine, and here we are getting 2 N-acetyl glucosamine attached to the dolicol, attached to the phosphate of the dolicol. So for this, when the N-acetyl glucosamine is added over here, we are using UDP, and in return we are getting one UMP and one UDP. Okay, let us see with the help of another diagram. See here, this is a dolicol, and attached to dolicol there are two phosphates over here. So whenever we are transferring an acetyl glucosamine, it is attached to UDP. Okay, so we have to attach to an acetyl glucosamine, so we need to UDP. Here first of all we are using the UDP, and in return we are getting one UMP and one UDP, which we have shown in the last picture. See here we are getting here one UMP, one UDP. In the first case, the energy is released and here one phosphate is attached, so that this UDP is converted into UMP. But in the second case, we don't need any of the phosphate, only this N-acetyl glucosamine needs to be transported. So this UDP is not converted into UMP. Okay, so only the N-acetyl glucosamine has been transported. So let us see in the next. Here one UDP is giving UDP and another one UDP is giving UMP. So this process takes place 2 times. That is the reason we have written here that it is 2x. Here we got 2 N-acetyl glucosamine. In the next step, we will see how this mannose is given over here. For the transfer of the mannose oligosaccharides, we need GTP. See here we are having here GTP. Attached to GTP, mannose is attached and it is transported. Here in the next step, mannose should be added, and for adding the mannose we need GTP, because attached to GTP there is mannose. Here there are mannose. Here this process will take place 5 times. So it is written here 5x. This is 1, 2, 3, 4, 5. One by one the mannose will be added. After the completion of the addition of the mannose in the cytosolic region, it will be flipped inside the endoplasmic reticulum lumen. This portion is the lumen portion. And as we have discussed that 9 mannose are added, but here we are having only 5, and inside the lumen rest 4 mannose should be added. So here for the addition we need another dolicol, which is a lipid molecule. Here it is a dolicol, this is a dolicol, attached to it there is a phosphate. And as we have to add the mannose, we need over here GDP, and attached to it there is mannose. This mannose will be converted, will be transported over here. Okay, these are the 4 mannose which are transported. How it will be transported? It will be transported one by one. At a time they will be not be transported. So when one mannose is coming here, the dolicol is bringing one mannose molecule. Again it is moving into the cytoplasmic area to bring another one mannose molecule. In this way, this process repeats 4 times. Okay, now we have completed with mannose. Now what we have to add? We have to add glucose now, and for adding the glucose we use UDP. Okay, because glucose is attached to UDP, and for this process again we need a dolicol, which is a lipid. This process takes place 3 times because we know that we are using 3 glucose molecules, so this process takes place 3 times. So In the first round, one dolicol will bring one glucose and it will attach and it will be flipped out. Again it will bring one of the glucose, again it will attach over here, again it will be flipped out. Again this process will take place. This process will take place 3 times. Now this is fully activated. This oligosaccharide is fully activated now. Now this oligosaccharide need to be transported. So where this should be transported? It needs to be transported into the protein and in some specific sequence of the protein. So what is the specific sequence? The specific sequence is aspargine, X, serine or threonine, and this X can be any of the amino acid except proline. Okay, as it is being added in the aspargine, it is known as aniline glycosylation because we can represent it as N. To the aspargine we can represent as N. That is the reason it is known as aniline glycosylation. So why do we need aniline glycosylation? Because the proteins need to be modified when it enters into the lumen, and after the modification only the folding of the protein will take place. This is all about today's lecture. In this video we have discussed about aniline glycosylation. In the next video we will discuss about the protein folding. Thank you for watching this video till the end, and if you like this video please do like, share and subscribe. Thank you.